I have given some lectures regarding cell biology, and now today I will explain you Golgi complex and lysosomes. First of all, Golgi complex. These are most developed in animal cells. And first discovered by Camillo Golgi in 1898. This is cell. This is nucleus, and let this be endoplasmic reticulum. I have already told you regarding endoplasmic reticulum. Now, Golgi complex have same structure as of endoplasmic reticulum, and actually the origin of Golgi complex is from endoplasmic reticulum. Golgi complex, it originates from endoplasmic reticulum, and the structure is same. There are curved stacks called cisternae, then small branchings called tubules, and fluid-filled small spherical structures called vesicles. That means all the structure resembles with the endoplasmic reticulum. The first part of Golgi complex is cisternae. These are few in number, placed in a heap, that means in a pile, one above another. But here, unlike endoplasmic reticulum, the cisternae are somewhat curved. So these bear two surfaces. This is convex surface, and this is convex surface, and this is their concave surface. Convex surface is also called cis-phase or forming phase. Cis-phase or forming phase, whereas concave one is called transphase. Or maturation. If you will take endoplasmic reticulum, there were also cisternae, but they were somewhat flat. Here these are somewhat curved. Then there are branching tubules here. These are tubules, just like this. These are tubules. And the most important function of the Golgi apparatus is to receive some proteins from the endoplasmic reticulum and convert or modify them into enzyme-bound small structures called vesicles. These are vesicles. And vesicles are usually filled with fluids. Those fluids are actually enzymes. The enzymes we usually call hydrolases. Hydrolases are actually our digestive enzymes. Our all digestive enzymes are called hydrolases. So, the main function of the Golgi complex is that they help in the formation of small membrane-bound structures called vesicles, and vesicles are filled with enzymes. So, these modify the proteins into enzymes, and when you will talk about enzymes, we say all enzymes are proteins. So, the proteins are received by endoplasmic reticulum. I have told you in previous lecture that endoplasmic reticulum have ribosomes over its surface, which are called rough endoplasmic reticulum, and the main function of ribosomes is to synthesize proteins, and from endoplasmic reticulum these proteins are modified or converted into enzymes by Golgi apparatus. So Golgi apparatus are mostly developed in animal cells, and there they help in the formation of enzymes. And if we talk of when after spermatogenesis, the sperm, here it gets converted into a structure called acrosome that also contains enzymes which helps in the digestion of egg membranes. This was Golgi complex. Now, the relative structure is lysosome. Relative structure is lysosome. I will explain you. Let this be a Golgi complex. These are cisternae with cis-phase and trans-phase. Then let this be a vesicle which has isolated from the Golgi complex, and it contains enzymes, hydrolysis, digestion. Now, a vesicle which separates from the Golgi complex, it is usually called primary lysosome. So, Golgi complex have the origin from endoplasmic reticulum. Lysosome have the origin from Golgi complex. It is called a primary lysosome. Now it contains digestive enzymes, and you know that the main this function of digestive enzyme is to digest food. So this is cell. Here the cell eats food, and you know that the food is taken through a process which is called endocytosis. Let this be food particle, food molecule or food particle. This primary lysosome, since it contains digestive enzyme, it has a property to digest the food molecule. These two combine, they form a session. A new structure is formed. Let this be Golgi complex, this lysosome, and this is food particle. Now this is called a secondary lysosome. Secondary lysosome. Now the food content is dissolved with the help of this enzyme. Enzymes help in the digestion of food particle. The material is transformed into energy by the cell itself, but some undigested matter remains here. So there is a remaining part. This is undigested matter. It is called tertiary lysosome. So we have seen three lysosomes. A primary lysosome, which is actually a vesicle which separates from the Golgi complex. Then primary lysosome, it forms association with food particle. It is called secondary lysosome. And secondary lysosome, the enzymes digest the food, and then some residue is left behind, or waste material is left behind, which is called tertiary lysosome, and it is excreted out. Here it was endocytosis. Here it is called now exocytosis. And in exocytosis, the residual body is thrown out, and it contains all the waste materials through exocytosis. And I have told you that there is a process called cell vomiting. So the main function of lysosome is to digest food. But sometimes there are some, for example, we will later on see that the cell contains mitochondria and other cell organelles. When they become aged, they become worn out, they are also digested by the lysosome itself. Let this be aged mitochondria. Now, and here primary lysosome forms association with its own cellular organelle mitochondria. A secondary lysosome is formed. It is also digested. It is the material can also be used for cell energy, and a waste material is formed. Here waste material is called autophagic waste. Since it is cell. So two types of waste materials are formed. One is residual body from outside source, and autophagic value from inside source, and it is also thrown out through exocytosis. And so important function is that it helps in digestion. It helps to remove the worn out material, old material, aged material from the cell. So it protects the cell. But one problem is here, that if a cell is not provided food, that means starvation is here. Starvation means food is not available for many days to a person, to an animal, and it first digests because food is not available. This part is not available at all. In the first instance, the worn out or aged material can be used, since the formation of lysosome is continuous process. Then what happens? The lysosomes then begin to eat live cell organelles. That means the mitochondria is functional, it is eaten. Endoplasmic reticulum is eaten, ribosomes are eaten. So it is the live content. That means it digests the live content and the cell dies. When such a condition occurs and when all the cells dies, a person can itself die. So these have been named as suicidal beings sometimes. That means these can kill a human being, these can kill an animal when food is not available, because these attack own organelles of the cell. So this was Golgi complex and lysosomes.